Okay, we're going to get started. Our first speaker is Dr. Farah Sheikh from UCSD, and she's going to talk about desmosomes, non-canonical functions in cardiac arrhythmias and disease.
Okay, thank you everyone. Thank you, Joe, for the introduction, and I'd like to thank the organizers, especially Drs. Zhang and Fukuda, for inviting me. So I'm going to talk to you guys about the cell-cell junction. I know it's been a hot topic for the cardiac repair field and the regenerative field, but hoping to kind of give you some insights into how these junctions work and what their biology looks like and how they can really interact and intersect with new types of diseases. So when we think about this desmosomes, we kind of have to look at the heart in much more detail and depth. And when you zoom in to two muscle cells that are tethered together, there are really three sort of junctional components that make up the cardiac muscle cell-cell junction. And desmosomes, that I've shown in this yellow box, these are really the mechanical anchors that couple adjacent cardiac muscle cells to one another, and they really resist high mechanical insult. And you can see that they're really coupled alongside gap junctions, which are important for synchronization of contraction, as well as the fascia adherens junction, which are coupled to the actin-myosin complex. Now, classically, when we think of diseases of the desmosome, we zoom in to the desmosomal complex and you see all of these proteins. 40 to 50% of cases are classically linked to this very detrimental disease called arrhythmogenic right ventricular cardiomyopathy. And essentially, mutations in each one of these genes is associated with this disease. And what's interesting about this disease is when you do get an effect at the desmosome, it sort of has this domino effect where now you're essentially destroying all of these other components. Most importantly, the gap junctions are oftentimes the first thing to be impacted. So I'm just going to give you some ideas about what is this classic disease that it's associated with, arrhythmogenic right ventricular cardiomyopathy. It's a complex genetic disease. It's historically defined, as you can see from its name, as a right heart disease, but recent studies identify left heart and biventricular involvement. And you can see also from its name that through electrical deficits really drive this disease. And you often see this can be made worse with exercise, and it leads to sudden cardiac death in young individuals. So there's this component that's clearly arrhythmogenic forward. But when you can look at the autopsies from these patients, there's clearly a structural component. You can see very little viable red muscle in this autopsied heart. Also, lots of fibrosis, as you can see in blue. And these circular cells are fat, and so they often get fat deposition as well. And what happens is that a majority of patients get heart failure. And this is also, interestingly, although a genetic-based disease, it's thought to be adult onset between the second and fourth decade of life. But desmosomal mutations are not just associated with ARVC. And clearly, you can see even when I described ARVC, there's this very complex syndrome where you get sudden death or you get structural disease. And when we look more closely at this, we can see that these mutations are often linked to also cardiac electrical diseases. So Brugada syndrome, they've been associated with both desmoplakin and plakophilin mutations. Also this particular syndrome, sudden unexpected nocturnal death syndrome, which also is found mainly in Asia, associated with DSP mutations. You can also get mutations in desmoplakin associated with long QT, as well as CVBT, which again, these are all arrhythmogenic and often have no impact on structure or function in terms of what we classically think of as mechanical dysfunction. So how do these proteins uniquely target ARVC, and how do they uniquely target electrical disease? That was sort of the question that we were really trying to understand. And so what we did is we did a molecular fishing expedition to find new desmosomal proteome regulators in the adult human heart. And we really focused on desmoplakin. It's in the N-terminus region, which is a hot spot for all of these interactions that it has with other proteins. And oftentimes a lot of pathogenic gene mutations are found here. And through this, we did a used-to hybrid screen using, again, the N-terminus region as a bait and using the adult human heart cDNA library. And through this effort, we identified a new protein called SNAP29 that interacts with desmoplakin. So SNAP29, when you look in the literature, not much is there in terms of the heart, but it's postulated to play a role in autophagy. It's thought to really mediate this autophagosome lysosome fusion step. And using studies in mouse dermal fibroblasts, it's thought to be a positive regulator. So when you lose SNAP29, it's thought to block autophagy and accumulation of autophagy markers such as LC32 and P62. Interestingly, it's also linked to this very rare human disease called Sedgwick syndrome. And this is sort of a disease that affects the brain, skin, and also has a component where it causes sudden death. And they actually don't know why the children die of sudden death. So that mechanism is unclear. So as I mentioned, no one really knows the role of SNAP29 in the heart. It remains undefined, and so we really sought to go after understanding it. And we hypothesized that it could be a new subcomponent of the cardiac desmosome that regulates desmosomal protein degradation because of its role in autophagy, and it may lead to sudden death. And so this is some studies that sort of validated this new protein. So you can see through this co-IP assay, when we pull down with SNAP29, we can pull down desmoplakin. We also looked at human heart sections via immunofluorescence, and you can see that SNAP29 is localized to the cell-cell junction alongside DSP, and you can see this merge, and this was also found in the mouse heart. And so this localization of SNAP29 at the cell-cell junction. And we used a desmoplakin knockout heart to sort of understand, interrogate its interaction. And so we looked at regions in the desmoplakin knockout heart where we still see cell-cell junctions, so we mark this with plakoglobin, and you can see in both wild type and knockout settings, the junction is there. However, we lose desmoplakin, we also lose a SNAP29 or vice versa. So you can see, unlike the wild type situation, suggesting that it could be a novel cardiac desmosomal interacting protein. And so to understand its role in vivo, we generated a SNAP29 conditional knockout model using XMLC2V Cree to knock it out specifically in the heart. And you can see from these Western blots that very early on when we lose SNAP29, there's really a selective loss of proteins. So desmoplakin is reduced, plakophilin 2 is reduced, which is often bound to desmoplakin, but one of the other things is connexin 43. So as I mentioned, this is kind of one of the first proteins that's targeted by loss of these mechanical anchoring genes. And you can see that the other parts of the cell really are not that impacted. But when we look at the physiology of the heart, it's very interesting. There's really no changes in cardiac morphology, and you can see from heart weight to tibia length. We also did Massen's trichrome stain. We really don't see any fibrosis. We looked at these mice, at least LV function over time at 3 months and 1 year, and really there's no differences in any of these parameters at this stage. So we actually did do some studies to look at arrhythmias in these mice, and this is where we saw phenotypes coming out. So a lot of, through telemetry ECG, lots of PVCs, a lot of electrophysiological deficits, and we put these mice under an invasive electrophysiological analysis and used isoproterenol as well as V stimulation as a means to sort of see if we see induced arrhythmias. And so in the presence of isoproterenol, at least in young mice, you can see that the SNAP29 knockout mice, a lot of them, up to 50%, undergo VTAC. But when we age these mice, we don't need isoproterenol anymore. In fact, all of these mice with V stimulation undergo electrical deficits associated with ventricular arrhythmias. So these mice really have an increased risk of baseline and inducible arrhythmias. Now, given that this is a mechanical anchoring gene, we sought to put mechanical stress onto these mice, and in the sense of we put them undergo transverse aortic constriction with pressure overloads. And essentially what happens is that this stress triggers ventricular fibrillation and sudden death. So essentially about 30% of the animals remain within this time frame after TAC. So this is really a gene that predisposes these mice to arrhythmias and sudden death. So far what I've shown you is that SNAP29 is a novel subcomponent of the desmosome. It contributes in the loss of function settings to ventricular arrhythmias in the absence of any structural heart disease. It also triggers, with mechanical stress, triggers sudden death. And so we were wondering, what are the mechanisms really governing the selective loss of these proteins as well as the arrhythmias in these cardiac-specific knockout mice? And so we did some TM analysis, and you can see autophagosomes really hovering around what we see are desmosomes. I hope you can appreciate this, but they're really surrounding them. We get an accumulation of these types of structures there. And also markers of selective autophagy such as P62 are increased, and in this case, without even any reporters, you can see the appearance of accumulation of LC3 at the cell-cell junction in these hearts. But we wanted to interrogate what are the real effects on autophagy, and so we isolated neonatal mouse cardiomyocytes from these SNAP29 flox mice and put in the setting of Cree to ablate SNAP29. And you can see here with loss of SNAP29 with the Cree, you can see again accumulation of P62 as well as LC3. And when we putting it in the setting of chloroquine, unlike what you see in non-cardiomyocytes, here now you get an accumulation even further of P62 as well as LC3B levels. And so we took advantage of Jun Sadashima's reporter, the RFP-GFP-LC3, and we looked in these cells to look at autophagosomes as well as autolysosomes, which will be essentially RFP positive. And this is just sort of a snapshot looking at the ratio of some of these autolysosomes to autophagosomes. And again, you can appreciate that this significantly increases autophagy activity and flux. So we just get so much more red. All of them are getting through to this step. And so if cardiomyocyte phenotypes driven by SNAP29 loss are due to increased autophagy, can we rescue these phenotypes with autophagy inhibition? And so these are some of the studies we did to show that, again, with loss of SNAP29, in baseline settings, you can see loss of desmoplakin, plakophilin-2, and connexin 43. And now when we do autophagy inhibition with chloroquine, we can increase the levels of these proteins back to LAC-Z levels. And also in the setting of baphyllomycin, you can see again, loss of SNAP29, loss of all of these proteins, and now we're able to increase the levels of all of these proteins back to endogenous settings. To look at the physiology of these cells, we exploited what's called the Excelligence, our real-time sort of cell analysis, and you can not only measure field potential, but contractility, and you can see again at baseline a lot of arrhythmias in these Cree-treated mice, which have loss of SNAP29, And again, with impedance, we sort of see similar deficits. But now, in the setting of bafilomycin and chloroquine, we now get restoration of the beat-to-beat interval that we see. This arrhythmogenic vulnerability that they have is no longer there. And so just to summarize, we believe that cardiomyocyte SNAP29 really insulates a subset of desmosomal proteins. And when we have loss of SNAP29, now these proteins are targeted for autophagy and essentially results in loss of these selective proteins, and that this provides this vulnerability to the cell for induction of ventricular arrhythmias, not only in vitro, as I've shown, but also in vivo, and that mechanical stress really triggers this even further. And so we believe that this insulation is important for this to restrict arrhythmias. So with that, I am happy to take any questions, and I'd like to first thank the Ji Wang, who was in my lab that sort of led all of these studies, Jun Sarashima for the use of his construct, and I'd like to thank my funding agencies, and happy to take any questions.
Questions for Dr. Sheikh?
Thank you very much for the beautiful data. I have one question. PKP2 and pathogenic variants sometimes cause the deficiency for the SCN5A trafficking. So did you check the interactions between ion channels, especially for the SCN5A?
Right, so we did immunofluorescence microscopy, and we do not see it changed in terms of its localization. We're now doing a little bit more nitty-gritty patch clamping, but again, those are isolated cells. They're not cells coupled, but so far we have seen no other changes in any of those calcium channels or any of the triggering, classic channels associated with arrhythmias.
Okay, thank you very much.
Mm-hmm.
Very nice talk, Farah. Just a quick question on SNAP29. I know it's involved also in endocytosis
Right.
and regulation of integrins, and I wondered if you looked a little bit in terms of its involvement in regulation of the integrin signaling pathways and how that might be regulating autophagy at the upstream level. Because I especially because it's in that tight junction, I
Right.
actually wonder how much of that is what's causing your phenotype as opposed to the autophagy alone.
Great question. We are looking at that now. I know it has effects on, trafficking and all of those things. So we are looking at integrins and also downstream signals of integrin. So we haven't looked at all of that yet. Thank you.
Thank you.
Farah, it's a very interesting observation. And so you're saying that the specific autophagy dependent destruction is locally inhibited, right?
Right.
So are you able to show co-localization of LC3 and desmosome component is increased?
So that's what I showed in that image. It might have not come across via immunofluorescence microscopy. So we looked at endogenous LC3, and it does co-localize to the junctional, to the, we use plakoglobin as a marker. And to be honest, it's not a complete knockdown of a lot of these proteins, so we can often see them like desmoplakin, plakophilin. So we often use that to co-localize. So I did see that.
Oh, great, fantastic, thank you.
Thank you, nice presentation. I want to ask a quick question about SNAP29. As you mentioned, cardiac knockout of SNAP29 induces ventricular tachycardia. And in these mutated mouse or humans, anti-arrhythmic drugs can be effective to prevent or stop these arrhythmia.
I have not tried, you mean like amiodarone or something like that? I have not tried the classic drugs that you would use in the clinic. So I have not checked that. But that's a great question. to look at that. I haven't used those.
Thank you very much.
No other questions? Thank you so much. Thank you.
Thank you.
Okay, so our next speaker will be my co-moderator, Dr. Masaka Aida. Dr. Masaka is the chair of Department of Cardiology at the University of Tsukuba, and I've been just told that he'll be the chair of Keio University Department of Cardiology starting June 1st. Congrats.
Thank you, Joe, for introduction. My name is Masaki Ida from now Tsukuba University, but I'm going to Keio University this June. And thank you, Jay, and thank you, Dr. Fukuda, for inviting me to this great symposium. So today I'm going to talk about transforming scar in heart muscle. No disclosure. As you know, so I don't think I have to explain this, but there is a high demand for the treatment of heart failure. And heart disease is a leading cause of death. And heart failure patients are increasing worldwide, and transplantation is insufficient due to shortage of donors. So in Japan, heart disease is a second killer, and mortality is like 200,000 population per year. So I'm working on cardiac reprogramming. So I'm explain briefly. So here is a normal heart, and there are many cardiac myocytes showing red, and many fibroblasts showing green. After MI, myocardial infarction, or cardiac injury, cardiomyocytes cannot regenerate, and resident cardiac fibroblasts proliferate, synthesize many extracellular matrix to make a scar in the heart. As you know, after that, cardiac function decline leading to heart failure. So our laboratory is focusing on direct cardiac reprogramming, converting fibroblasts into cardiomyocytes without making iPS cells. So this is a kind of abstract of my work, and we previously found that overexpression of 3 cardiac transcription factors, GATA4, MEF2C, and TBX5 in short GMT can reprogram fibroblasts into cardiomyocytes. And we also show that using Sendai virus vector-mediated GMT overexpression can reprogram resident cardiac fibroblasts into cardiomyocytes in vivo in acute MI, which was published previously. So I think most of you know cardiac reprogramming is effective in acute MI in mouse. However, there is an important question. The effect of cardiac reprogramming in chronic MI remained unknown. So chronic heart failure is an unmet clinical need. So our laboratory is focusing on chronic MI in this paper. So today I'm going to talk about cardiac reprogramming could improve cardiac function and reduce fibrosis in chronic MI, which was published very recently in Circulation. We found that overexpression of 4 cardiac reprogramming factors, MEF2C, GATA4, TBX5, and HAN2, could reprogram fibroblasts into cardiomyocytes and improve function and reduce fibrosis. Mechanistically, we found that profibrotic cardiac fibroblasts exist in MI, established scar, can convert into cardiomyocytes. It's kind of regeneration. But fibroblasts can kind of convert into quiescent type of fibroblast by overexpression of MGT through inhibition of MEOX1, which is a master regulator of kind of bad fibroblasts. So reprogramming, regeneration, and anti-fibrosis can improve cardiac function. So to test cardiac reprogramming in chronic MI, we generated triple transgenic mouse here, TCF21I3, cross tomato, and MGTH2A mice. Because as you know, mouse is very small, so of course we can do viral vector injection in acute MI, but in chronic MI, it's very difficult and challenging for technical reasons. So that's why we use tamoxifen-induced expression of MGTH and tomato in resident cardiac fibroblasts by using TC21, which is a specific cardiac fibroblast-specific clearline. By administration of tamoxifen, we can label resident fibroblasts by tomato, and also simultaneously we can overexpress cardiac reprogramming factors in the fibroblasts, and we expected we can make new muscle cells in the heart. This is very important. We realize that there are many tomato-positive cardiac fibroblasts 3 months after MI, as shown here. So as here, there are many fibroblasts even after 3 months. So these cells are kind of target of cardiac reprogramming in chronic MI. So this is a kind of strategy of in vivo cardiac reprogramming in chronic MI. We generated MI by coronary artery ligation in the mouse, and after 1 month, we administered tamoxifen in the mouse for 5 days, and then reprogramming started and continued for 2 months. And after 3 months, we harvested the heart and checked cardiac reprogramming. We compared control and triple transgenic mouse. Here is the data. And we found that in the control mice, very, very few cells expressed cardiac troponin T, of course, and there no regeneration. But in triple transgenic mouse, some tomato-positive cells started to express cardiac troponin T. And as shown here in the inset, the cells express sarcomeric structures. The reprogramming efficiency was not so high. It's like 22% in the border zone in triple transgenic mouse, but we could generate new cardiac muscle cells from fibroblasts by overexpressing cardiac reprogramming factors in chronic MI. We next analyze ICM more closely. So here, and here is a tomato-positive induced cardiomyocytes, which express connexin 43 gap junction and coupled with surrounding cardiomyocytes. We isolated endogenous cardiomyocytes and induced cardiomyocytes, as shown here. Of course, endogenous myocytes didn't express tomato, but the ICM expressed tomato and cardiac troponin T, and as you see, the morphology is pretty similar. So these results suggest that in vivo ICMs could couple with surrounding cardiomyocytes. Next, functional data, we did echo as shown here, and after MI, the ejection fraction was reduced from 60% to like 30%, and then after 1 month, we administered tamoxifen to induce cardiac reprogramming in chronic MI, and then we found in the reprogramming mouse, the ejection fraction was increased, but not in control mouse. We also analyzed fibrosis because we thought that fibrosis kind of fixed and cannot be changed after 1 month or 2 months. But surprisingly, we found that fibrosis was reduced or recovered after cardiac reprogramming, as shown here. So this is the area of fibrosis, and as you see here, cardiac reprogramming could reduce fibrosis or reverse fibrosis in chronic MI. Next, it's very interesting that we found reversal of fibrosis in the established scar. So we wanted to know the mechanism of anti-fibrosis by cardiac reprogramming. To test this, we use single-cell RNA sequencing in non-muscle cells. We isolated non-myocytes from sham, control MI, and triple transgenic mouse ventricles, as shown here. And as you see here, there are many types of cells, including fibroblasts and cellular cells, etc. And this is the integration of 3 groups. And as you so these yellow cells are fibroblast population. And as you see here, compared with sham, in the fibroblast population in control MI was increased, which was reversed in triple transgenic mouse. So this is a ratio of non-myocyte cluster, as shown here, and the fibroblast population was increasing MI as expected, but this was reduced after reprogramming. So these results suggested that cardiac reprogramming could reduce CF population. Next, as you know, fibroblasts are not homogeneous. We think it's heterogeneous populations. So we analyze more closely about cardiac fibroblast population. So here is a single-cell RNA sequencing of fibroblast population. They express collagen 1A1 homogeneously, but COMP and periostin, which are marker of profibrotic fibroblast or ECM secreting fibroblasts cited in the left side. And this is a cluster analysis of cardiac fibroblasts. So CF5 is a kind of marker of profibrotic cardiac fibroblast population. This is the data of sham, MI, and reprogramming mouse. And as you can see here, as shown in the red arrows, the blue CF5 population was increased in control MI, but this was reduced in triple transgenic mouse, suggesting cardiac reprogramming could reduce profibrotic CF population. So this is a frequency of CF cluster. So as you see, CF5 was increasing in MI, but reduced in triple transgenic mouse. So these results suggested that cardiac reprogramming can change the fibroblast cell state. Next question is that why and how cardiac reprogramming can change CF cell state. So to determine this, we analyze RNA velocity analysis as shown here. So in the control mouse, MI mouse, the CF cell state didn't change much, but after reprogramming, the CF5 population was moved or transit to the right side, which is a quiescent type of cardiac fibroblast. So these results suggested that profibrotic cardiac fibroblasts convert into quiescent type of cardiac fibroblast by cardiac reprogramming. We test this kind of more quantitatively by pseudo-time analysis, and as you can see here, the triple transgenic mouse show more broad expression of colors, suggesting this cell state is changed in triple transgenic mouse. So these results suggested that cardiac reprogramming converted profibrotic CF to a quiescent state. It's a kind of conversion of fibroblast state by cardiac reprogramming. Next question is that why cardiac reprogramming can change CF cell state. I think many of you may know MEOX1 is a marker or master gene of bad fibroblast or fibrosis after myocardial injury. So we tested, for example, fibrogenic transcription factors may be regulated by cardiac reprogramming. So this is a volcano plots, volcano plots of single-cell RNA sequencing in cardiac fibroblast. And red indicates sham, and green indicates control MI, and blue indicates triple transgenic mouse. And interestingly, we found that only MEOX1 was increased in control MI, which was reduced in triple transgenic mouse, but SNE1 and also SOX9, both of them are kind of known as a fibrogenic transcription factors, didn't change much, suggesting MEOX1 might be important for fibrosis in chronic MI and also may regulate antifibrosis induced with cardiac reprogramming. So next, to determine the role of MEOX1 in cardiac reprogramming, we performed an in vitro experiment as shown here. We use alpha-myosin heavy chain GFP transgenic fibroblasts in which only cardiac myocytes express GFP, so fibroblasts don't express GFP. And we overexpress MGTH, cardiac reprogramming factors, plus MEOX1 to restore the MEOX1 expression after reprogramming. We expected some of cells express alpha converted into ICM, but many of them remained as cardiac fibroblasts. We FACS analyzed cardiac induction by alpha MCGFP expression, and we also analyzed fibroblast state by sorting alpha MCGFP negative fibroblasts and did QRT-PCR analysis to investigate the fibroblastic state in the cells. This is the results, and of FACS analysis, we found that in the control of no GFP, but after overexpression of MGTH, 8% of the fibroblasts started to express GFP, suggesting the reprogramming efficiency was 8% in vitro, but this was reduced or suppressed by overexpression of MEOX1, as shown here. So MEOX1 suppressed MGTH-induced ICM generation. Next, we analyze fibroblast population. As shown here, overexpression of MGTH suppress MEOX1 expression as shown in vivo, also in vitro. And overexpression of MEOX1 restores expression of MEOX1. And as you can see here, collagen 1, COMP, and TGF-beta 1, which are all hallmarks of fibrosis, were increased after overexpression of MEOX1. So these results suggest that MEOX1 suppress MGTS-induced antifibrotic effects. So in summary, we found that MGTH induced antifibrotic effects through suppression of master regulator of fibrosis, MEOX1. So this is a kind of summary of our paper. We found cardiac repairment could repair chronic MI, and we found the mechanism of this repair. Lastly, I'm going to talk shortly about our new data of how we can improve cardiac repairment efficiency. So in this paper, we perform in vitro experiment mostly, and in this case, we overexpress activated MEF2C in combination with GATA4 and TBX5 to generate more ICMs. So here is activated MEF2C, which had mildly transactivation domain in the C-terminal of native MEF2C. And as you can see here, the number of beating ICMs were greatly increased by overexpression of activated MEF2C with G and T. And interestingly, we found that ICM induced with GT plus activated MEF2C had more adult cardiomyocytes like phenotype, which is which means we can make more mature type of cardiomyocytes by using activated MEF2C. Next, we analyzed RNA sequencing, and we found that it's very small, sorry, and the ICM generated by activated MEF2C are more similar to adult cardiomyocytes compared with native MEF2C with GT, with GATA4 and TBX5. And sarcomere length and also cell size were also increased with M2 plus activated domain, as shown here. And carbon-3, which is a marker of T-tubule, is expressed only in activated MEF2C with GATA4 and TBX5, but not in normal MEF2C. And also electron microscopy reveals that GT plus activated MEF2C had more mature type of cardiomyocytes having a T-tubule formation. So activated MEF2C could generate more mature ICMs as shown here. Next question is that how activated MEF2C could improve cardiac reprogramming. To determine this, we did chip atlas search for regulatory factors. We found that in the more than 1,000 genes upregulated in M2 activated form, and in these candidate factors, of course, MEF2C, GATA4, TBX5, NKX, these cardiac transcription factors are recruited into the MEF2C bounding site, but we found that EP300, P300 is also can be also recruited to this cardiac low site. So this is a chip analysis, seq analysis in the upregulated genes in the activated MEF2C with GT, and we found that P300 was recruited, can be recruited to the same location as MEF2C, GATA4, and TBX5 with active enhanced epigenetic marker. So this is a kind of example, NAPR and TMOD1, which is cardiac specific genes. So P300 was recruited here. So this paper was accepted in stem cell reports and will publish soon. And lastly, I'd like to acknowledge members in Tsukuba University. They help a lot to me, and thank you very much for your great attention. Thank you.
Questions for Masaki? Roberto, you have a question? Go ahead.
Yeah, very nice work in the chronic model in the mouse. Are you working to bring that to a larger model where you can actually inject these three factors? So have you tested this in larger models of chronic ischemic cardiomyopathy?
Sorry, I cannot catch. So did you say that using mouse model or a pig
No.
model?
Yes, are you using a large model like big model to
Yes.
test these three factors that you tested so elegantly in the transgenic mice?
Yes, I think so, but we didn't test it yet. Yeah, I would like to do that in the future. Thank you.
Thomas?
Very nice work indeed. Is that one? Yeah. Two questions actually. So one, I mean, there have been also reports that a cocktail of small molecules, so Wnt inhibitors, HDAC inhibitors, butyrate, and tretinoic acid, I think also enhances such transdifferentiation. So I wonder whether you also try to use that in vivo to augment your model and transdifferentiation.
Thank you very much. We didn't test chemicals in vivo so far. I think other groups did, so I don't know how effective it is because we don't have experience.
And
But
second question,
yeah.
I mean, I found this really interesting that you have, on one hand, effects promoting de novo generation of cardiomyocytes, on the other hand, attenuation of fibroblast activation. So if you would just judge the impact of these two different processes, so what matters more to reduce scar formation? Is that the suppression of fibroblast activation or conversion to cardiomyocytes? So what's more important?
I think both important, of course. But of course, the efficiency of direct cardiac reprogramming is still low. So I think I would like to improve cardiac reprogramming, than making
The effects
good
are
fibroblast.
probably more due to attenuation of fibroblast activation, so in general, probably.
Yeah, thank you.
Let me ask you about the last part of your talk. You said when you use the MEF2C plus transactive activator domain, you said the P300 was enriched in the upregulated genes, but is that because of the activating domain?
Thank
because it can recruit P38 anyway.
Thank you. So I didn't show the data, but we found that inhibition of P300 inhibited cardiac reprogramming. So that's why we think that P300 recruitment was critical for improving cardiac reprogramming.
Thank you.
Thank you. We also did chip analysis. Thank you.
That was a great talk. So I'm curious why you choose TCF21 CRE to do the reprogramming. Maybe you can choose like parallelstein because that is for the activated fibroblast. So do you see different like reprogramming efficiency for the quiescent and the fibroblast and the activated fibroblast?
Sorry, you said that what type of fibroblasts are more easily reprogramming into cardiomyocytes?
Yeah, because
activated
after
or
MI
quiescent?
Yeah,
Is that right? Okay. We couldn't test because TCS21 is a kind of homogeneous expression in fibroblast population, so we cannot discriminate activated and quiescent fibroblasts using TCS21. So to answer your question, maybe we should use periostin ER3, but we didn't test it.
Do you see the reprogrammed cells on the remote zoom?
Sorry?
Did you see the reprogrammed cells on the remote zoom?
In remote area? No, actually, very few, yeah.
Okay, thank you.
Yeah, so should be maybe activated a little bit. Yeah, thank you. Thank you.
Good. Thank you so much.
Next, I'm going to introduce last speaker in this session. His name is Job Van Vero, Associate Professor of Medicine, Director of Graduate School in University of Minnesota. Please start. Thank you.
Thank you. I'd like to echo the other speakers and thank the organizers for inviting me. I realize I am the last speaker to stand behind or before the end of this first day, which I think was a great day. So I'll try to keep it short and to the point. I'll talk today about endoglin, which is a TGF-beta co-receptor. Of course, everybody knows that injury induces cardiac hypertrophy, ultimately leading to heart failure. The way I look at this is when you have a loss of cardiac myocytes indicated by the gray areas on the left, the rest of the myocardium needs to compensate for this, and that ultimately leads to dropout of additional cardiomyocytes, making your therapeutic interventions with drugs less and less effective. And I think the best way to counteract that would be to increase the cardiomyocyte content of the heart, as we have been talking about all morning. As you all know, the difficulty is, of course, that during embryonic development, proliferation is very abundant in the first 5 days. That's the main thing that happens. And afterwards, you have a lot of differentiation to give rise to the individual organs. But after birth, there is a complete lack, although exactly at what time point in humans this happens, we don't really know. But cardiomyocyte proliferation comes to a stop. In mice, this has been elegantly shown by Lauren Field in the past, that it happens a couple days after birth, about 4 to 5 days after birth. You have a complete block of cardiomyocyte proliferation, or virtually complete block. And the question is, can this be reverted, yes or no? And we've heard a couple of talks suggesting that indeed this can be reverted. This is very different from lower organisms like newt and zebrafish, where adult cardiomyocytes can de-differentiate, become proliferative to the point as needed for organ repair, and then re-differentiate into mature cardiac myocytes. And of course, we have all heard today that the SEDAC laboratory at UT Southwestern has shown that this can also happen in mice, and later groups have shown that this can also happen in pigs. But it can only happen in the first 1 or 2 days of life, because at that point, cardiomyocytes stop proliferating. And if you don't have cardiomyocyte proliferation, you don't have regeneration. By now, we know a lot of genes that can affect cardiac myocyte proliferation, but we hear more and more papers coming out every day that additional regulators of cardiac myocyte proliferation might exist. So to tackle whether we could identify additional regulators of cardiomyocyte proliferation, I started a project when I was still a postdoc in the lab of Jeff Mulkenton, where we performed a genome-wide screen to identify novel regulators. This was done in cell culture initially, and today I'll talk about one of the hits coming out of that initial screen. So the screen was done using fetal mouse cardiac myocytes isolated at embryonic day 17 and a half, when in vivo they are still proliferative. We pre-plated these cardiomyocytes for 1 hour to remove most of the fibroblasts, and then infected them with a lentivirus that expresses a short hairpin RNA. And then 3 days later, we added EDU for 24 hours to measure S-phase in cardiomyocytes. When you plate cardiac myocytes 1 day after plating, you can still see much proliferative activity within cardiac myocytes. This goes down quickly at day 2, and by day 3 after plating, there's virtually no cardiomyocyte proliferation in these cardiac myocytes anymore, suggesting that even though in vivo they're still actively proliferating by day 19, when you take them into culture, this dissipates quickly. And the question then is, can you inhibit specific genes to reactivate that potential that was still present in vivo? We use scrambled shRNA as a negative control. You can see virtually no EDU-positive cardiac myocytes there, and we used adenoviral MYC overexpression as a positive control, where about 50% of the cardiac myocytes become EDU-positive. We enlisted the help of Jeff Saucerman at the UVA to help with automated image analysis of these millions of images. And he helped us with identifying individual cardiomyocytes and then discerning whether they were EDU positive or not to score individual genes, whether they were pro-proliferative or not. When you do this at this scale in an automated manner, you get a lot of positives, and many of these turned out to be false positives due to the algorithm. So there was verification done for all the positive, potentially positive hits by eye to make sure that indeed the algorithm called it correctly, and only the blue ones are true positives. So here's an example of the negative control, the scrambled RNA, the positive control, the MYC overexpression, as well as 4 potential or 4 hits coming out of the screen. CDKN1A is well known as P21. This is a cell cycle inhibitor. RBL1 below that is also a cell cycle inhibitor. So these are well-known regulators of cell cycle, and I was very pleased to see that we identified some of these well-known cell cycle regulators. And then we identified additional regulators like MEPRI3, which is a microtubule end binding protein, as well as RAB8A, as potential regulators, novel regulators of cardiomyocyte proliferation. And they all stimulated EDU incorporation, but not all of these actually also progressed to G2M as marked by phosphorylation of histone H3. We then tried to verify in vivo whether inhibition of some of these genes, the most prominent ones, would actually give rise to new cardiomyocyte formation. And unfortunately, the vast number of genes that we tested did actually not give rise to cardiomyocyte proliferation in vivo. And we don't really understand why there is such a difference between the in vitro results versus the in vivo results, but presumably this has to do with the maturation status of the in vivo cardiomyocytes, probably. But the one hit I want to talk about today that we did see stimulation of cardiomyocyte proliferation is a TGF-beta receptor called endoglin. Endoglin can bind to TGF-beta as well as BMP receptors and modulate how these receptors respond to TGF-beta binding or BMP binding. And this is well known to regulate proliferation in endothelial cells, for example, and the exact mechanism of endoglin seems to be very much cell context dependent. So this is in mouse cardiac myocytes. When we inhibited endoglin using shRNA, we saw about a 2-fold increase in EDU incorporation and also about a 1.5 to 2-fold increase in phosphorylation of histone H3. When you look at endoglin expression on cardiac myocytes, isolated cardiac myocytes, this goes up from P1 to P7 to adulthood. So we thought, okay, maybe inhibition of this gene could be a promising target for restoring the cardiac myocyte proliferative ability. But instead of going first for adults, we stuck with relatively young neonatal mice. We used endoglin floxed mice, crossbred them to the cardiomyocyte-specific inducible Cre, gave tamoxifen 1 day after birth, and then 6 days later performed apical resection, and then injected EDU 6 days after apical resection, and then measured EDU incorporation within cardiac myocytes that are marked by PCM1 staining. And we saw a significant increase in the number of cardiac myocytes that were positive for EDU. There was about a 2 to 3-fold increase from about 8% to about close to 25% of cardiomyocytes that were EDU positive. They were not just EDU positive, many of these were also positive for phosphorylated histone 3, about 4% of cardiac myocytes, and we saw some evidence of cardiomyocyte cell division as indicated by aurora kinase B staining. So does this then result in actual cardiac repair? To measure this, we first looked at cardiac function, which was increased in mice that were deleted for endoglin. I'm sorry that the label is missing. Oh, here is the label. And we measured scar formation, which was significantly decreased when we inhibited endoglin on cardiomyocytes specifically. So this, I think, is really exciting that we could inhibit endoglin and then thereby stimulate cardiomyocyte proliferation and increase cardiac repair. But as you may know, endoglin is actually highly expressed on non-cardiomyocytes, specifically fibroblasts and endothelial cells. And some of that endoglin is actually cleaved and becomes soluble. So the question is then, if we inhibit endoglin on fibroblasts, is that also sufficient to stimulate cardiac myocyte proliferation. So to be able to test that, we crossed the anaglin flux mouse to the TCF21 tamoxifen-inducible Cree to delete it from cardiac fibroblasts. And again, performed apical resection, similar strategy as for the cardiomyocyte-specific Cree. And we did indeed see an increase in the level of EDU positivity of cardiac myocytes after apical resection, but the level of phosphorylated histone H3 was not significantly increased in these mice, suggesting that even though there is some activation of cell cycle, it is not sufficient likely to give rise to no cardiac myocytes. And that was also reflected in inhibition of increase in ejection fraction or fractional shortening. However, when we then looked at scar formation, this was actually completely, well, maybe not completely, but significantly blocked when we inhibited anaglin on fibroblasts. So that suggests that scarring could be separate from cardiomyocyte proliferation. And I think that is an interesting thing that we would like to interrogate further later on. So far I've shown you that anaglin inhibition stimulates cardiomyocyte proliferation in cell culture in mouse and rat, and that cardiomyocyte-specific anaglin deletion actually enhances cardiomyocyte proliferation and cardiac regeneration, while fibroblast-specific anaglin deletion blocks scar formation but does not stimulate true cardiomyocyte proliferation. So I told you that anaglin is a regulator of TGF-beta signaling. So of course we looked at whether TGF-beta signaling is affected by using cultured cardiac myocytes inhibiting anaglin, as shown in the second bar, where we see an increase in EDU positivity of cardiac myocytes. And then when we, on top of that, inhibited additional regulators of the TGF-beta signaling pathway, like TGF-beta receptor 1 or TGF-beta receptor 2, we saw indeed that anaglin likely signals through the TGF-beta receptors, but when we inhibited SMAD3, we did not see any effect. And this is in contrast to when we inhibited SMAD2, where we saw that the pro-proliferative effect of anaglin inhibition was dissipated when we also inhibited SMAD2. And this was further confirmed by using a adenoviral dominant negative SMAD2, where we saw a dose-dependent inhibition of the pro-proliferative effect of anaglin inhibition. So these data suggest that SMAD2 is required for the proliferative effect observed after anaglin inhibition. So it's all nice to show this, of course, in mouse, and we wanted to verify whether this was actually also active in human cardiomyocytes. So we used iPS-derived cardiomyocytes and again used siRNA to block anaglin and then measure EDU incorporation within cardiomyocytes. The baseline EDU incorporation rate in our cardiomyocytes was relatively low, about 3%, and this went up to about 16 or so % in the anaglin-inhibited cardiac myocytes, with again some evidence of cell division as evidenced by aurora kinase B staining at the cleavage furrow. We then isolated RNA from these human cardiac myocytes and performed RNA sequencing, and you saw big changes in gene expression. On the left are the control siRNA, and on the right anaglin siRNA, and when we then did PCA analysis, there was good separation between the control and anaglin siRNA treated cardiac myocytes, and when we then finally did pathway analysis, it was interesting to observe that the upregulated pathways with anaglin inhibition were all related to mitosis and chromatin, while the downregulated pathways with anaglin inhibition were all related to sarcomere expression or cardiomyocyte, cardiac muscle function. So that suggests that maybe anaglin inhibition suppresses the cardiomyocyte differentiation states to allow for cardiac myocytes to become proliferative. So with that, I would like to conclude that anaglin is indeed an inhibitor of cardiomyocyte proliferation, at least at the early neonatal stages, and deletion of anaglin can enhance cardiac regeneration after apical resection, and the proliferative effect requires SMAD2 signaling. I think for us, the next steps would be to test whether this is still possible in adult mice, and we have not yet begun to assess that. But I think that will be a critically important next step. With that, I would like to thank the people who did all of the work, especially Daniel Sorensen, who was a graduate student in the lab and has since moved on to do postdoc at Stanford, as well as Doba Yusul, who also was a graduate student in the lab, has since moved on to do postdoc at Boston Children's, and Jeff Saucerman, as well as my funding sources. Thank you for your attention.
Thank you. Any question or comment from audience? Please.
Yeah, super interesting, really. Was wondering, did you also look for SMATs related to BMP signaling, so 158? Is there any change in phosphorylation?
We did also assess whether inhibition of the BMP receptor as well as BMP9 could be modulating the endoglin inhibition-induced proliferative effect. And to be honest, the results were confusing. When we did this at baseline, inhibition of SMAD1 or SMAD5 actually reduced the cardiomyocyte proliferation rate, and this was blocked when we had endoglin inhibition on top. So probably TGF-beta signaling and BMP signaling have potentially opposing effects on cardiac myocytes, and I think we have to do a better job at precisely interrogating the contribution for both of these. But it's well known that endoglin has very pleiotropic effects, and that makes it challenging to perform reductionist experiments and then broadly conclude how it works.
And we overexpress the BMP10 transiently in the heart, yeah? And surprisingly, this induces a massive atrophy. I mean, similar things have been described, constitutive expression of BMP10 from your colleagues there from Indianapolis, but surprisingly, if you do that in the adult heart, you also induce a massive atrophy. And I was always wondering whether endoglin signaling would also contribute to that. But
that is to be determined. We're breeding the mice, and I hope to be able to start those experiments soon.
Thank you.
Thank you. Back.
Very nice, Jaap. Beautiful, elegant work. My question is very similar to what you just said with regards to endoglin regulation from cardiomyocytes and crosstalk to other cell types, and whether you've looked to see if there were paracrine effects that actually mediated some of the functions. And so I'm surprised that you actually didn't see effects. You saw differential effects from cardiomyocytes to the fibroblasts, and that you didn't see any kind of contributions from cardiomyocytes to fibroblasts or fibroblasts to cardiomyocytes that were similar. And I guess sort of following up on that question is, did you look to see if there were regulatory signals through RhoA signaling, which I know affects limb kinase, not so much on the proliferation side, but more for fibrosis? And then I noticed that you were doing a lot of apical resection studies. So have you looked, probably you already said this, but I missed it, but have you looked in the adults after injury to see if you can recapitulate some of that proliferation / fibrosis effects that you talked about?
Yeah, great questions, Maria. Thanks. We have not yet looked at Rho signaling, and we also have not yet looked in the adults, but really important studies that we have to do. I think another experiment that we have to do probably is mixing cardiac myocytes and fibroblasts and have one endoglin deleted and the other not, and testing the cross talk between them. You probably know that there is an antibody that can block soluble endoglin. We contacted the company that produces this. They did not have a mouse version available anymore, but they were allowing us to use the human version, TRC-105. And when we use this on human iPS-derived cardiac myocytes, we saw some minor effects, but it wasn't very strong, suggesting that membrane-bound endoglin is probably the endoglin that matters for stimulating the for inhibiting the proliferative capacity of cardiac myocytes. Okay. But I think we have to do more in-depth studies to really nail this down.
Okay. Very nice. Thank you.
I have a quick question. Yeah, great talk as always. So on your iPSL studies, in which you took the cardiomyocytes and you add endocrine, and you show that there's an increase of BRDU, and that you interpret as cardiomyocyte proliferation. I think for the BRDU uptake, it could reflect DNA damage. It could reflect dedifferentiation. It could reflect proliferation of the cardiomyocyte. It could also reflect proliferation of the contaminant endothelial cells or contaminant fibroblasts. So I wonder, have you done actual FACS sorting of troponin-positive cells to show that there's an increase in your troponin-positive cells rather than just by BRDU uptake? Yeah.
That's a great question. When we measure EDU incorporation, we specifically only measure this in troponin-positive cells. So we only look at the cardiomyocyte specific EDU uptake. And we show that that also coincides with increases in phosphorylated histone H3, as well as increases in aurora kinase B staining at the cleavage furrow. So all of this was increased when we inhibited endoglin. But we have not yet actually counted whether that leads to an increase in the number of cardiac myocytes in the dish. I think that's a good suggestion.
Good. Okay, no other questions. Thank you so much for this outstanding talk. I'm not sure if Kaiichi or Jay going to make some conclusion comments. You guys want anyone?
Thank you very much for a wonderful conference today. From now, we will have a gala dinner at 7 o'clock. That will be held at the Mizuho Room at the Westin Kyoto Miyako Hotel. All the invited speakers and their families and abstract presentators and some other guys can attend this gala dinner. So please come together before 7 o'clock. That room will open 6:30. Thank you very much.